Welcome to the latest Novogene webinar, Next Generation Sequencing and its Clinical Applications. Today's webinar focuses on how recent advances in sequencing technologies are being applied to understand, prevent, and treat human disease. All our previous webinars, including our ever-popular Beginner's Guides series, are available to view on demand on our website. A bit of housekeeping before we start. The presentation will run for around 40 minutes with 15 minutes for question and answer at the end. Please submit your question via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We will try and prioritize the most popular questions. If we don't have time to answer your question, we will come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any question via the Q&A or message us directly if you have a technical issue. We will share the slide deck and a recording of the webinar via email. You can expect this on Friday. If you are studying and need a certificate of attendance, there will be a link in the follow-up email that will display the certificate, which you can print from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this, as helps us to plan future webinar topics. I would now like to introduce your host for the webinar, one of our Spanish account managers, Oriol. Oriol, over to you.
Thank you, Amanda. Hi, everybody. So thank you for joining today's webinar about exploring next-generation sequencing and its clinical applications. So my name is Oriol. I'm Novogene's account manager for Portugal and the host for today's webinar. So shortly, I will pass over to Larissa, who will talk you through the clinical potential of NGS. But before I do that, I'm going to speak a little bit about Novogene, especially for anyone who's joining us for the first time, and what we can offer you to assist and help in your next-generation sequencing journey. So Novogene was founded in 2011 and has been growing rapidly since then. And our company's mission is to advance genomics and improve life. And as you can see here, our services cover a huge range of applications with real-life implications, from complex disease research and oncology to food and water safety. So our European Sequencing Center is based in Cambridge in the UK, in the Cambridge Science Park. So here we have our state-of-the-art lab equipped with the latest high-throughput Illumina machines, the NovaSeq 6000 sequencing machine. We have always provided fast turnaround times and high-quality data, but that is set to improve even further with the installation of the Falcon 2, which is our new intelligent sequencing platform, which I will touch on further shortly. And we also have our supercomputing cluster, so we can even offer bioinformatics service and also store your data in the UK. However, at heart, we're a global company, so we can offer services across Asia and the US, and of course, Europe. And with almost 2,000 employees, and we're already serving more than 5,600 customers in world-class universities, biotech companies, and hospitals. And we can provide sequencing from the smallest to the largest of projects. So we provide full range of sequencing services, such as whole genome sequencing, whole exome sequencing, various types of RNA sequencing, and even further, epigenomic services and metagenomic sequencing services. And as you can imagine, this can be applied to a wide range of research topics, from human disease, microbial cell factories and gut health, to agriculture and animal health. So the focus of today's webinar is on our clinical service, which comprises sequencing of genome, transcriptome, and epigenome. So altogether, it can help building a complete overview of the disruptions happening in the patients, and thus providing a mechanistic explanation of the disease. So the NGS market is crowded, and there are certainly a lot of good reasons why you should trust Novogene with your project. So we have had a really exciting development recently, as I said before, with the installation of the Novogene Falcon 2, which is our new intelligent NGS delivery platform. So this new technology was developed in-house by Novogene and will massively increase the capacity of our Cambridge Sequencing Center. It will also increase the quality of the data produced, since it's entirely automatic, thus removing any possibilities of human error. And with this new addition, we are now able to even speed up and offer our fastest ever turnaround time for RNA sequencing and whole genome sequencing of just 10 working days from sample arrival to data release. And this Falcon tool really highlights the innovation we strive for here at Novogene in an effort to keep pushing the frontiers of genomic research. So the benefits of this, what I mentioned, this in-house innovation are clear in our record in the scientific community, which we are really proud of. We have been cited in over 790 research papers with a total impact factor of more than 6,000. And at the same time, we're also driving the NGS industry forward, since we have over 37 NGS related patents. And our technology that is helping to make the NGS process more efficient and effective, and all the while improving the quality of the data we deliver. So, and as a proof of what I just said, I just would like to show you my own publication where we took advantage of Novogene's RNA sequencing and pre-made library sequencing services, which together with their bioinformatic pipelines provided us with, importantly, ready to publish results. And that certainly helped us to speed up both the research and the publication of these results. So the Novogene community keeps on growing. Our expert sales team speaks over 10 languages, and we are located across Europe, ready to help and assist you in your next project. So even if you're not ready to send us samples yet, you can still benefit from our educational outreach to learn how NGS can work for your project. And actually, we recently wrapped up the first installment of our European roadshow, where different members of our sales and product teams delivered seminars about the applications of NGS in DNA and RNA research. So please keep an eye on our social media for future seminar announcements that hopefully we will be visiting near you soon. And with this, I'd like to introduce our lovely sales team who will be answering your questions. And we're, as I said, located all over Europe. So first we have the UK and the Ireland team, followed by our DACH team and our Benelux region team. But our team has grown so much lately that it takes 2 slides to cover them all. So here I'd like to introduce our Southern Europe team, the Nordic and the Eastern European teams. So if you would like to speak to your local sales representative about your upcoming NGS project, please contact us via our website and you will hear back from us and from your local sales person. And with this, I would like to thank you for your attention, and I will now hand over to Larissa, who has got a master in genetics and is one of our product marketing managers at Novogene. And within that team, she's in charge of our clinical sequencing products. And with her expertise in genetics, she will guide you through the applications of next generation sequencing into the clinics. So over to you, Larissa.
Thank you very much for the nice introduction, Oriol. And also my big welcome to our webinar today. Thank you for joining, and let's get started. So first of all, let me introduce today's topics. So first, we're going to briefly touch on the evolution of technologies and their principles. Then we're also going to look at some multi-omics approaches, including from genomics, transcriptomics, and epigenomics, and how these methods can be applied in clinical research. Finally, we're also going to take this into practice, introduce some clinical applications to human diseases and how this can be translated into personalized medicine. And at last, I'm also going to introduce a few other relevant applications. So let's start off with the question of what is next generation sequencing, or NGS. So NGS is now the current generation sequencing. It is more time and cost efficient than standard and has fundamentally changed the questions we can ask and answer as scientists. So thinking about genome sequencing, the first draft of the human genome was published in 2001. And since then, there has been considerable advances in the field. This is actually really nicely summarized in the Nature Milestone article. And if you're interested, please feel free to have a look. It highlights the evolution of more advanced sequencing platforms, which we're also going to talk about in the next few slides. For example, in 2007, the development of ChIP-seq, which combined chromatin immunoprecipitation with high-throughput next-generation sequencing, enabled the genome-wide interrogation of chromatin binding patterns of different proteins, giving insight into regulation mechanisms, development, and epigenetics. Or in 2009, long-read sequencing technologies began to shed light on hidden parts of the human genome. So generally speaking, there are 3 generations today. So Sanger sequencing is the process of selective incorporation of chain-terminating D-deoxynucleotides by DNA polymerase during in vitro DNA replication. So then you can visualize everything on the electrophoresis gel. It is a low-throughput sequencing method and is still widely used. So I'm assuming that most of you actually are quite familiar with this method. Generally, Sanger sequencing is able to sequence 500,000 base pair fragments. Hence, it's a short-read sequencing platform. Illumina, on the other hand, is a massively parallel sequencing technology that offers ultra-high throughput sequencing scalability and speed. It is the most used platform today, and it also sequences 50 to 500 base pair fragments. So both Sanger and Illumina are considered short-read sequencing tools. And recently, a 3rd generation has been developed to sequence long reads. So for familiar companies are probably Oxford Nanopore and PacBio. And they basically sequence native DNA in real time with single molecule resolution. And this way you can sequence tens of KB fragments on average. And next generation sequencing is actually referring to the 2nd and the 3rd generation sequencing tools. So let's go through some of the principles. So the Illumina is based on a sequencing by synthesis principle, and it uses reversible denominators that enable the identification of single bases as these are introduced into DNA strands. So it basically reads fluorescence after each letter is added, and then a fluorophore is removed, which allows more adding. PacBio, on the other hand, is based on a single molecular real-time sequencing principle, and it captures sequence information during the replication process of the target DNA molecule. It also reads fluorescence as letters added. Instead, Oxford Nanopore is slightly different. It measures the changes in current as a base passes through the pore. We are also now briefly touching on the different sequence modes of PacBio. So PacBio has the continuous long read mode, or the CLR mode, which basically generates 1 pass on each molecule sequence. Therefore, it is able to sequence very large incisors. However, if there's a base error, this is not corrected. On the other hand, circular consensus sequencing, or the CCS mode, generates multiple passes on each molecule sequence. And therefore, if there's a base error, it can actually be self-corrected, because you get a consensus read at the end, which is also known as a hi-fi read. And here I just want to compare the different sequencing platforms again to give you an overview on what is best in your situation. So the key factors here are actually accuracy and read length. So as already said, Illumina is only able to sequence very short reads, and therefore is limited to the novel assembly and also to sequence structural variants. However, it is relatively cheap and it's very accurate. It's also the most widely used application. And PacBio and Oxford Nanopore instead are both able to generate really long reads. However, they are quite error prone. PacBio has the advantage that it has the CCS mode, which is actually really accurate. Whereas the CLR mode, as mentioned in the previous slide, is not that accurate. Both PacBio and Oxford Nanopore are able to generate directly detect epigenetic modifications. However, they are quite expensive, and you also need quite large amount of starting material for the library preparations. So since now we have familiarized ourselves with the NGS technology, we can move on to look at some multi-omics approaches. So I want to start off with whole genome sequencing, which is basically where you're trying to obtain a comprehensive picture of an organism genome. So we're not going to spend that much time on these. These are quite standard in terms of approach and service, but generally there's 2 different approaches here. So you have novel sequencing and resequencing. The novel sequencing is the approach where you want to sequence a novel genome where there's no reference sequence available yet for alignment. So your aim is here to assemble and annotate a reference genome. On the other hand, resequencing is where you sequence a species where a reference genome already exists. So here you really want to compare the differences between genomes from the same species. However, I want to point out that the human genome is very well studied and most advanced in DNA sequencing. So you wouldn't really do any de novo sequencing anymore because there is a lot of databases available, such as ClinVar, Omnium, Cosmic, etc., to compare the variants. And generally, a whole genome sequencing is able to detect most variants. So they can be grouped into 3 different categories. We have single nuclear variants, which are 1 base pair change. There are indels, which include deletions and insertions, and they're smaller than 50 base pairs. And we also have structural variants, which are bigger than 50 base pairs and include tandem duplication, interspersed duplications, inversions, translocations, and copy number variants. So whole exome sequencing, on the other hand, is actually focusing on coding regions of the genome. So why are researchers interested in WES? Because when you sequence only 2% of the genome, you're actually able to identify 85% of known disease-related variants, which makes it easier to detect rare and low-frequency mutations because the coverage on the target is increased. It is simply a cost-effective alternative to WGS. And whole exome sequencing consists of 2 steps. So first, you want to select only the subset of DNA that encodes proteins, which is known as the exome enrichment. And different companies provide different panels of probes. So for example, here at Novogen, we offer Agilent, IDT, and Twist. And if you need more information about these different panels, please feel free to contact your local account manager after the webinar. And once you've done your exome enrichment, you can actually sequence the external DNA using high-throughput DNA sequencing technologies. And this is also a great moment to introduce our newest kit for exome sequencing, the Twist Exome 0.2. So the Twist Exome 0.2 is designed for human genome 38 and specifically optimized for NovoSeq instruments. However, most importantly, it has the superior coverage of major genetic databases such as RefSeq, MCCDS, GeneCode, ClinVar, and many more. And the addition of clinically relevant non-coding pathogenic and likely pathogenic variants provides the value of multiple clinical panels all wrapped into one. And there's a lot of information about in this slide. However, I'm not going to go through it in detail. And again, if you have any more questions, feel free to contact us after the webinar. So target region sequencing makes everything more focused. So it really only focuses on a subset of genes or regions known or suspected to be associated with a disease or phenotype under study. So you can detect known and novel variants within a region of interest by requiring less sample input and producing a smaller, more manageable data set. So there are 2 different approaches here. So you can do hybridization, which is quite similar to whole exome sequencing. So you're basically designing probes to target regions. And here at Novogen, we have the NovoPM 0.2, for example, which entirely focuses on cancer targets. And then amplicon sequencing, on the other hand, is slightly more affordable and easier. So you would definitely amplify a region of interest out, and you would sequence it after. So it's generally used for detecting CRISPR editing events, genotype and sequencing, and other applications. And here on this slide, we're just going to compare the 3 approaches again. So WGS generally looks at all genes and known coding DNA, including structural variants and mitochondrial DNA. Hence, this one is getting most information from this method, but also a lot of data which needs to be handled. WES, on the other hand, optimized cost per sample, but you're not able to detect any alteration in non-coding regions, which are known to also play important roles in gene activity, protein production, and hence can lead to diseases. It, however, makes you able to achieve comprehensive coverage of coding variants, such as single nuclear variants and indels. It is not able to identify large structural variations, however. Target region sequencing is best to use if a certain disease wants to be investigated. It provides precision without driving up sequence costs to achieve the required depth of target regions. However, you wouldn't use that for any broad discovery research. And the depth you require would really depend on your project aim. So although WGS and WES have tremendously improved our understanding of the genetic etiology of human disorders, about half the patients still do not receive a molecular diagnosis. So compared to DNA, which remains nearly identical across all cells and time, the transcript was very dynamic. So RNA sequencing improved diagnosis through both illuminating the functional consequence of variant of uncertain significance and helping in the prioritization of variants either prioritized by WJS or detected by WES. So RNA sequencing is valuable for interpretation of coding as well as non-coding variants, and sneaking clinical practice is getting bigger and bigger. So let's look at some aberrant RNA phenotypes. So aberrant expression, for example, can be identified as defined as an expression that significantly deviates from the normal physiological range. And chromosome packaging, histomodification, RNA polyadenylation, all tightly regulate gene expression. And genetic variation, both coding and non-coding, is well known to impact these processes. So a low expression level of a disease gene often translates into a low protein level, indicating a genetic disorder. So RNA sequencing is also used to detect monoallelic expression, which refers to the situation where mainly one of the two alleles is expressed, while the second allele is only expressed at very low levels. And in about 2% of WS and WG as unsolved cases, monoallelic expression of a disease gene can be detected, leading to a molecular diagnosis. So the last one I want to discuss is aberrant splicing. So it's been estimated that around 94% of human genes undergo splicing, and the majority of them actually have alternative splicing. So this is regarded as a key cellular process in ensuring functional complexity in higher eukaryotes. And it's estimated that at least 10% of pathogenic variants have an effect on RNA splicing, whose effect in splicing is hard to be predicted based on the DNA sequence alone. Therefore, RNA sequencing plays an important role in directly probing splice isoforms and adding a functional layer to a variant detected by WGS and WES. So splice defects could result in exon skipping, exon truncation, exon elongation, exon creation, and intron inclusion. So let's add another layer of complexity. So epigenetics comprises regulatory mechanisms that do not affect the DNA sequence, but it can alter chromatin compaction to modulate gene expression. So DNA methylation is what occurs when methyl groups tag DNA to activate or repress genes. And histomodification occurs when the binding of epigenetic factors to histone tails alters the extent to which DNA is wrapped around histones and the availability of genes in the DNA to be activated. And all these factors and processes may result in cancer, autoimmune disease, mental disorders, among other illnesses. And it can be affected by several factors and processes, including development, chemistry, and drugs. And they offer an opportunity to improve disease diagnosis and to predict clinical outcomes. And epigenetic modification can now be widely analyzed using NGS. For example, DNA methylation can be analyzed using whole genome bispecific sequencing. Histomodification and DNA protein interaction can be analyzed using ChIP-seq. You can look at non-coding RNAs by RNA sequencing, and you can also look at RNA protein interaction using RIP-seq. So here I actually want to introduce to you some PacBio human genomic solutions, as provides the most comprehensive view of genomes, transcriptomes, and epigenomes. It improves rare disease research efforts with hi-fi sequencing to unlock the biology of rare diseases that's inaccessible through other technologies. So hi-fi sequencing enables simultaneous phasing of reads into maternal and paternal haplotypes and detection of methylation through measuring the kinetics of nucleotide incorporation. And the Sequel systems directly output long and accurate hi-fi reads with annotation of methylation at all CpG sites. And the advantage here is that there's no library preparation, special library preparation needed like bisulfite treatment. And PacBio can therefore drive personalized approaches to medicine and care with high-resolution insights into medication response, adverse drug reactions, and decreased efficacy. It can also achieve complete and deep full gene HLA profiling without any fragmentation. And it can identify true HLA allelic diversity for improved transplantation in immunological research. And more of that actually later. I'm going to talk about that a bit more later. So since we've now looked at all the different multi-omic approaches, let's look at some clinical applications. So before we actually start digging into the different analysis approaches, I would like to show you some how genomics impacts clinical care from before birth, but to even after death of a human being. So genomics can really be useful at any stage in your life. For example, reproductive carrier screening is carried out before pregnancy or in early pregnancy, and it can be used to identify a couple's chance of having a child without a serious or with a serious genetic condition. And post-mortem testing can identify a cause of a death individual who may have died suddenly or unexpectedly, and it can therefore help to prevent the same outcome in surviving family members. But really, you can see here that you can come across genomics throughout your whole life. And diseases generally fall into two categories according to their genetic architecture. So we have monogenic diseases, and as the name says, they are mostly caused by a single gene with typical 1 to 2 variants per family. And a Mendelian inheritance pattern can be observed. Again, more on this later. And common complex disorders, which tend to have many causal variants with small effects, and they can be influenced by environmental factors leading to a phenotype, and hence they are polygenic. So the spectrum of disease-causing variants is based on frequency of the allele and its effect. And the minor allele frequency is defined as alleles positive for the variant over total alleles screened. And generally, a common variant is over 1% or 5%, and rare one is under 1%. And Mendelian diseases actually fall into the upper left corner here on this graph, having a rare frequency variant with large effect. And traditionally, linkage mapping was used to combine genotypic and phenotypic data to obtain the chromosomal location of interest before targeted sequencing. But now linkage analysis is more combined or integrated into the analysis of the sequencing directly. The sequencing is mostly family-based. Common diseases, on the other hand, fall into the lower right corner, having common variables with small effect. And association studies are the way to identify those. Rare variants with small effects are extremely hard to identify, whereas common variants with major effects influence common diseases. So let's now have a look at some numbers concerning rare diseases. There are more than 7,000 known rare diseases, and more are discovered every single year. However, fewer than half of them actually have resolved genetic etiology. And around 300 million people suffer from a rare disease, and around 80% of these are genetic or at least have a genetic component. And NGS increases the likelihood of rare disease diagnosis and provides the fastest path to end this diagnostic disease. However, you can see on this picture here that it still takes an average diagnosis of around 8 years. And some common rare diseases are actually Gaucher disease, Hunting disease, cystic fibrosis, and many more. So as already said, Mendelian diseases are mostly caused by mutation in a single gene and transmitted according to a Mendelian pattern of inheritance. So after variant detection and exclusion of variants that are unlikely to be causal, one probably identified many pathogenic variants or likely pathogenic variants or even variants with unknown significance. So, but how can you filter them more? For example, here at Novogene, we provide you with a filtering based on a disease model to narrow down the set of variants. So let me introduce some of the inheritance patterns to you. So an autosomal recessive pattern means that an affected individual requires 2 mutant alleles in the same gene to be affected. In autosomal recessive patterns, you would expect a compound heterozygote, meaning the presence of 2 different mutated alleles at a particular gene locus. The parents would also each have 1 mutated allele being heterozygous. In consanguineous families, you would expect the same mutated allele at a particular gene locus, hence a homozygous variance, because the parents are somehow related. However, I do want to point out monoallelic expression again, because heterozygous variants alone are often discarded when investigating autosomal recessive disorders. But when only 1 allele carrying a rare deleterious variant is expressed, and the other allele is nearly completely silenced, the variant behaves like a homozygous variant on the transcript level. And this phenomenon can only be detected by RNA sequencing and can be due to genetic or epigenetic factors. Then X-linked recessive works slightly different. So the mutation is on the X chromosome, meaning boys are generally more likely to be affected, whereas females are likely carriers. And autosomal dominant refers to disease which require only one mutant allele to cause the phenotype. Hence, he would investigate heterozygous variants in affected members. This can be quite challenging when the family is quite small, because you will get a lot of different candidate mutations which you have to filter further. So linkage analysis is actually another BI or bioinformatic approach to increase the ability to identify causal variants. So it's the co-segregation of a chromosomal region and a trait of interest, and relies on using family-based data to detect genetic loci that may harbor disease predisposing genes. And as you can see here, children 1 and 5 are recombinants, which narrows down the region of interest to the 3rd and 4th loci because these are not linked to these regions, and they're both affected. And the LOD score is a statistical test that compares the probability of obtaining the test data if the two loci are linked to the probability of obtaining the test data purely by chance. So linkage analysis can provide you the location of the variant. It can be a measure of strength of the evidence in being involved in disease predisposition inherited in a particular pedigree, and it gives you information on haplotypes. And as already pointed out on the previous slide, dominant disorders may reveal many candidate variants. So if you can actually delimit the region of interest by linkage, the location with respect to the linkage interval can help you to reduce the number of candidate variants to a more manageable size. And at Novogene, we offer a non-parametric linkage analysis based on called single nuclear variants and single nuclear polymorphisms. So new mutations have long been known to cause genetic disease, but their true contribution to the disease burden can only now be determined using family-based WGS and WES approaches. So a de novo mutation is a new germline mutation caused by mutagenesis occurring in parental gametes that's not inherited from either parent. Such a genetic variant occurs for the first time in one family member, and thus forming the first generation of carriers, who can then transmit this new variant to their offspring. However, it has to be said that many dominant Mendelian disorders occur sporadically, and due to severity and early onset, they are not transmitted. So therefore, new insights have come from studying these sporadic forms by NGS using family trees or quartets. There's also mosaic de novo mutations, which do not appear in the whole organism. And here you really want to compare different tissues. So since now we've talked quite a lot about rare diseases, let's move on to common diseases. So these are generally more complex, as these are influenced genetically, but also by our lifestyle. And they are one of the most pressing challenges today. They rely on many common genetic variants with small effect on the phenotype, and which then determines the genetic architecture. So genome-wide association studies have enabled us to scan hundreds of thousands of genetic markers. It is used to identify genomic variants that are statistically associated with risk for disease or particular trait. So in GWAS, allele or genotype frequency are compared between individuals with the disease, the cases, and unaffected individuals, the controls. So in the Manhattan plot, which is shown here, an association signal typically results in a peak, arising from regional correlation of a genetic variation caused by linkage equilibrium among SNPs. And linkage equilibrium is the non-random association of alleles of different loci. And again, GWAS has successfully identified variants in diabetes, Crohn's disease, Parkinson's disease, and many other diseases. So again, this is another great moment to introduce another application, which is low-pass WGS, or also known as low-coverage or shallow WGS. So this basically means that you learn from less data by sequencing at low coverage, relying on imputation to fill in the missing information. And the advantage of this approach is that it's cheaper, and you can identify new rare variants, which is not possible with traditional genotyping arrays. So one application is in fact GWAS, but it can also be applied to evolutionary studies and many others. So since now we have covered rare and common diseases, let's move on to some cancer applications. So cancer genomes are unique with different genome alterations, for example, point mutations, copy number variations, structural rearrangements, and others. And in fact, though normal human genomes are mostly alike, every cancer genome is different. So this quote actually describes the book well, which says, all happy families are alike, each unhappy family is unhappy in its own way, which can be kind of directly translated to cancer genomes. And even though we've already touched on this slightly before while talking about rare diseases, I want to define somatic and germinal mutations again, just because it's a very relevant concept for cancer. So germinal mutations are heritable changes in the germ cell lineage. Therefore, they can be inherited to the offspring, and they affect the entire organism, and therefore they can also affect evolution through natural selection. And you would really sequence a blood sample to detect those. On the other hand, somatic mutations, they occur in somatic tissue of an organism, and they can result in a mosaic individual. They cannot be inherited, and they're known as acquired mutations. And in fact, these are the ones which are the most common cause of cancer. And the influence is local, and you would basically sequence tumor samples and compare the results to normal counterparts. So what information can we actually get from NGS? So for example, we can look at driver mutations. These are mutations that provide a selective growth advantage and therefore promote cancer development. They're also known as active mutations, and they're a good target during cancer therapy. So mutations without this impact, they are termed passenger mutations. So understanding this helps us to understand the genetic roots of how cancer cells form and differ from healthy cells. And cancer cells actually accumulate somatic alteration over time. So mutations may be acquired while the cell lineage is phenotypically normal, reflecting both intrinsic mutations acquired during normal cell division and the effects of extraneous mutagens. And during the development of the cancer, other processes, for example, DNA repair defects, may contribute to the mutational burden. And passenger mutations do not have any effect on the cancer cell, as we've mentioned before, but driver mutations are the ones which cause the real clone expansion. And relapse after chemotherapy can also be associated with resistant mutations, which often predate the initiation of treatment. And the mutations frequency actually depends on the cancer type, and external forces of the drug treatment can select for specific clones. So in fact, an important concept to consider when investigating cancer is tumor heterogeneity, which is when different tumor cells show different morphological and phenotypic profiles. And understanding the influential factors is crucial, as it is a huge challenge to personalized cancer treatments. So for example, we have intertumor heterogeneity, which is a genetic and phenotype variation which can be observed between tumors of different tissue and cell types. But there's also differences between individuals of the same tumor type. And we also have intertumor heterogeneity, which occurs within a tumor, where subclonal diversity can be observed. And in fact, tumor cells can also show heterogeneity between their expression profiles. So it's important to identify gene expression signatures and mutational profiles. And single cell RNA-seq and even spatial transcriptome-seq are more advanced approaches and very useful here. And we've actually briefly touched on this in the previous slide, but Peter Noble, he actually proposed that cancer arises through an evolutionary process, and he published a landmark reflective piece on that. So he proposed that cancer arises through stepwise somatic acceleration mutations, which inevitably leads to having multiple subclonal populations. So another way of looking at mutation is through TMB, tumor mutational burden, which is a clinical biomarker. So what is TMB? So it refers to the number of genetic changes in a cancer cell and is reported as the number of mutations seen in a section of DNA and reported as mutations per megabase. And TMB varies by cancer type, exposure, age of onset, and DNA repairability. And non-synonymous mutations result in amino acid change in the protein, which result in new shapes, which is known as new antigens. These are considered to be foreign to an immune system. Therefore, immune checkpoint inhibitors, or also known as ICIs, are able to stimulate and allow the immune system to detect these new antigens and destroy the tumor. And cancer with low TMB have actually fewer mutations and this decreases the chance that the immune system is activated. Whereas cancers with a high TMB have more mutations, therefore increasing the chance that at least one will activate an immune response. So let's now look at some testing approaches used to identify damage with therapeutic options, screening, and early detection, which may be important for you when you choose what method you would like or need to use for your own studies. So there are two main biopsy approaches here used to detect somatic mutations of cancer: tumor biopsy and liquid biopsy. So tumor biopsy is actually looking at all mutations in the DNA of cancer cells and has the advantage that it's high sensitivity and a standard procedure. However, it is quite invasive and it is not representative of tumor heterogeneity and evolution. Liquid biopsy, on the other hand, looks at small pieces of DNA that are shed by cancer cells into the bloodstream, and it has the advantage that it's non-invasive and it can detect heterogeneity and evolution. However, it has the disadvantage that it's lacking a standard, as it's a relatively new method. Germline testing, on the other hand, uses blood to detect germline mutations, which may be associated with increased risk of different cancers, for example, Lynch syndrome or BRCA1 and 2 genes, which can inform screening, result in early detection, and allow prevention. However, please be aware that blood cancers and bone marrow patients, transplant patients, cannot use blood samples. Therefore, you would need to use something else, for example, a skin biopsy. So finally, here we can show you the road from NGS to personalized medicine. So personalized medicine is the tailoring of disease treatment or management to the individual characteristics of a patient. So the approach relies on scientific breakthroughs in our understanding of how a person's unique molecular and genetic profile makes them susceptible to certain diseases. And the same approach is increasing our ability to predict which medical treatments will be safe and effective for each patient, and which will actually not be effective and should not be used. So finally, I've introduced to you some other relevant applications. So the first application I want to introduce is HLA genotyping, which is the identification of HLA class 1 and 2 gene polymorphisms for individuals. So the HLA system actually encodes the major histocompatibility complex proteins in humans, which is responsible for the regulation of the immune system. And it consists of mainly two classes, whereas class 3 proteins are located between class 1 and 2, and they're not thought to be involved in antigen processing and presentation. And the HLA gene complex is actually located on a 3.6 MV region within chromosome 6. And the HLA genes are the most polymorphic gene family found in the human genome, with more than 10,000 different HLA alleles reported to date. And these genes have been strongly implicated in transplant rejection, autoimmune disease, vaccine pharmacogenomics, cancer, and many others. Therefore, HLA genotyping is applied in organ transplantation, population evolution, as well as immunological disease and cancer studies. And the development of NGS has actually completely changed this landscape of genotyping compared to traditional HLA genotyping and Sanger sequencing. Because it enables robust, simple, high quality, and high throughput analysis of the key HLA genes, and the data can actually be phased to a minimum of 6 digits. And here we can use either WAS, WGS, or even direct capture approaches. And finally, let's explore some mitochondrial genomics. So mitochondrial DNA is inherited exclusively from the mother, and the disorders are heterogeneous and can arise any stage of life. And heteroplasmy is a well-known phenomenon, meaning that mutant mitochondrial DNA and wild-type mitochondrial DNA coexist, which can influence an individual's phenotype. So, for example, a normal phenotype can be observed when there's quite a high range of wild-type mitochondrial DNA, whereas a mutant phenotype is observed when there's a high amount of mutant mitochondrial DNA. And you can apply mitochondrial DNA sequencing to study human diseases, but they're also important for human identification and forensic application. And again, here you can use whole genome sequencing, but also whole exome sequencing with specific spike-ins for mitochondrial, and you can also have specific target panels just entirely for mitochondrial DNA. So in conclusion, there are three main approaches on the road to personalized medicine. Genomics is used to identify variants potentially causing a phenotype, and transcriptomics to identify different differential expression patterns, alternate splicing, RNA editing, and finally epigenomics, looking at the effect of methylation and histone modification and their effects on gene expression. And the functional effect of a mutation with various environmental factors in a network or pathway needs to be analyzed in order to develop strategies to prevent and even to treat diseases. So now we've reached the end of this webinar, and I would give the word again to Oriel for starting the Q&A session.
Thank you. Thank you, Larissa, for this brilliant and informative presentation. So it's really clear, I think, for everybody that next generation sequencing has really advanced our knowledge of the underlying mechanisms of the disease, and that this complexity really needs to be addressed from multiple points of view, like from the genes themselves, how the genes are altered by either mutations or epigenetic modifications, and overall how these end up affecting gene expression. And it's what's also really nice to see as well is at the very moment, like we can address all these points by multiple sequencing approaches. And I'm really happy to see how this is and how this will slowly change clinical practice. So with this, I would also like to start the Q&A, asking one question myself, and it is if you could elaborate more on the specific advantages of long-read sequencing for whole genome sequencing, if the entire genome has already been sequenced for many years, and specifically what you meant in your presentation when you said NGS dead zones.
Yeah, thank you very much for the question. And in fact, I will actually go back to the slide where we talked about PacBio human solutions. And as you can see, the advantage basically of PacBio is the long reads. So the advantage here is that you can sequence and cover multiple regions which are inaccessible. So for example, this could be repetitive regions which are quite hard to identify through short reads, just because you cannot sequence them very well and you don't really get overlapping regions while assembling them, which you do when you do long read sequencing, because you get more overlaps. And dead zones could be, like I already said, can be some repetitive regions or just some regions which are really hard to detect through short read sequencing. However, the advantage here is also that you can directly detect epigenetic modifications. You don't need to do another library preparation as you have to do with short reads. So you can directly detect two different concepts and do two different analyses, which is very helpful and easy to do. You're also able to look at parental imprinting, because you can face the haplotypes directly. So it has a lot of different advantages, and recently there's also been shown that you can sequence telomeres much better, which was not really working with short reads. So there's lots of different hidden parts in the genome which are much more or easier to sequence with long reads. Yeah, hope this is a bit clearer now.
Thanks, Larissa. It was indeed really clear. Following with PacBio, we have another question from Ramin, who says that to clarify, when you mentioned high accuracy and high error rate, when talking about PacBio, and another question he had is whether the PacBio continuous long read mode, the CLR setting, if it's inaccurate with high errors and long reads, would it be advisable to use Oxford Nanopore instead?
Let me just quickly go to that slide again, just because it's easier to see. Yeah, so far I understand the question correctly is then why would we still use CLR mode if it's not really that accurate? So sometimes you can use the CLR mode if you, for example, want to do a de novo assembly, and you want to basically sequence using short reads, and you just want to use the PacBio to kind of do hybrid assembly, for example. So you just use these longer reads to find to be able to assembly a genome better. This is and it's where you can use that approach basically. However, if you want to completely sequence a new genome just by using PacBio and you want to look at structural variations and other aspects, I would always recommend to you to use the circular condensed sequencing mode. And yeah, regarding nanopore actually has the disadvantages that I can go back to this one, and that is actually really high error prone as well due to systematic errors with the polymers, which you can see here. And also the disadvantage is that there's actually quite a lot of changes in the software and kits. So there's not that much material on it yet to for the different analysis. So it's yeah, it wouldn't be really used instead of PacBio in my opinion. So yeah.
Thanks, Larissa. I think it's really clear. So now jumping from long read sequencing, we have some questions regarding target region sequencing instead. And Lola asked if you can target sequence like an entire chromosome, let's say 50 to 60 megabases instead of the whole genome.
And yes, you can certainly do that. And as I said, you have to basically here. So you can basically have different approaches here. You can look at, if you want to maybe just create a new panel where you can entirely only target the region of interest. However, this may be quite might involve quite some time, and also it may actually be more expensive depending on how big the genome is you want to sequence. Or you can also do some amplicon sequencing, maybe where you can just try to amplify the regions and sequence those amplicons. It could be another approach. But generally, yes, you can. You don't have to sequence the whole genome. You can also look in generating your own panel, for example. With Agilent or or some else company.
Thanks, Larissa. Actually, we have another question regarding target region sequencing from Suraj, and he's asking if copy number variations, TNVs, can be detected using targeted region sequencing.
No, you would not use this approach to look at copy number variations. You can still detect some copy number variations using whole exome sequencing. However, if to get the full picture of all different structural variants, I would always recommend to do whole genome sequencing, because this is targeting everything. Whereas the more target you get, the least you can actually look at longer structural rearrangements, including copy number variations.
Thanks, Larissa. I think it's really clear the advantages of whole genome sequencing. And now, jumping from TRS, Peter is asking regarding whole exome sequencing, and his question is, which are the advantages of using Twist 2.0 instead of the other kits for whole exome sequencing?
And yeah, so I actually have this whole slide where you can see what all the different benefits are, but it's actually also Twist 0.2 is actually able to capture the 2 strands of DNA, and therefore it increases the capture efficiency. It is a very high coverage and uniformity, and the most important thing is actually embedding non-coding regions. So you can also look at all of these non-coding regions, including the TAB promoter, which, for example, is not covered by the Agilent V6 version. And so this is basically, you have all these different regions or like contents which can be analyzed, which basically summarize a lot of different kits all into one.
Thanks, Larissa. We can now jump from genome to more multi-omics approaches, and Ramin has just asked whether the discovery of the importance of the non-coding and long non-coding RNAs, if this increases the importance of the transcriptomics over proteomics. What's your opinion?
Wait, can you just repeat this question? So if the long non-coding RNAs and transcriptomics are more important than genomics, or?
No, Dramin is asking whether due to now increasing knowledge about the non-coding regulatory molecules like long non-coding RNAs, microRNAs, if this increases the importance of transcriptomics over proteomics.
And I think, yes, I mean, the transcriptomics generally provides you with this functional layer. So you can see which genes are, genes are more likely to express and how this reflects directly into proteins. So of course, the more a gene is expressed, and which is completely regulated by non-coding RNAs, such as long non-coding RNAs or small RNAs, and they actually influence how much of a protein is going to be there. And this is definitely a very important approach to see if what the different disease patterns are. So transcriptomics is definitely one of the most important approaches here. Yes.
Yeah, I completely agree with you, Larissa. So now jumping more from transcriptomics to epigenomics, Paloma is asking us about methylation, and actually she would like to know if we include methylation of studies in cell-free DNA at the moment, or if we are planning to do it.
So we are actually, we are always working on improving our protocols, of course, and we are, so like I said, we are already providing the, if you're doing PacBio, for example, you can look at at methylation and at the DNA sequencing at the same time. So this is one approach where you can basically look at to get both information. And it also kind of depends on your project and what your aims are, just to kind of tell you what approach we should use and what product we would offer for you to you. So I would actually recommend that we contact you after this webinar again to discuss this a bit further. Because yes, there's loads of approaches we can suggest for you to see how you can include both, especially also for cell-free DNA.
Thank you, Larissa. And I think now this question was really nice to introduce the next topic, and one which is really important when talking about clinical samples, which is the starting material. And the first one we have is from Alex, and he would like to know more about the input for the different platforms. And he's asking, like, for example, if you're working with small input samples like circulating tumor cells, what options do we have available for them?
Yeah, of course, there's different material has different, you're more likely to get very fragmented and also degraded material for especially patient samples. So for example, FFPE and even cell-free DNA can be low concentrated. Therefore, of course, we have different input materials for these different starting materials. And you can also look at that when you look at our starting material or the sample requirement guide, where we can list the different material, the different requirements for each of the services, and how, of course, we always optimize our protocols to also accommodate your low input samples, for example, because I know that cell-free DNA can be quite hard, or it can be very hard to get loads of material, such as for genomic DNA. But of course, we can also talk to you about your project after this and see how much we can, how much you need. But of course, yeah, lower amounts are definitely possible.
Thanks, Larissa. Indeed, I mean, this is really nice introductory questions, because a lot of people are also sending us FFPE samples. And Betty is asking us actually if there's any problem when analyzing mutations from this kind of samples.
So yes, already said, basically if you're able to get enough material and the quality of the material is sufficient according to our requirements, again, these are of course adapted to accommodate that FFPE samples are generally just more degraded. And then we are definitely able to provide you with the same kind of results than with genomic DNA. There should be no problem about that.
Thanks, Larissa. And now kind of the last question regarding the starting material from Jin Kui. So he is asking us whether, to getting qualified libraries that from samples that have really low biomass, maybe PCR cycles could be a solution. And he's actually asking us whether that would lead to low accuracy when sequencing these samples.
And yes, of course, you do have to pay attention if you amplify your DNA or your amplicons too much, because then you may get some PCR duplicates, which may not be helpful for the analysis later. However, therefore, yes, it's always good to enrich low quality or low amount samples. However, if there's always a limit to that, so it shouldn't be overdone, let's just say that, because then you would just not get good results after anymore.
Thanks, Lisa. And now I would just like to conclude the Q&A session with another question from Ramin, in this case regarding whole genome sequencing. And it's a really interesting question. And it is whether if you could expand more on the importance of whole genome sequencing compared for cancer genome profiling and its advantages over whole exome sequencing, considering that it is maybe a little bit more inaccurate.
You mean, so whole genome sequencing is generally used because we can see that more and more that the non-coding regions are actually also very important to detect how cancers evolve and how cancers occur. Therefore, whole exome sequencing, whole genome sequencing is the approach to basically get most information. However, it does still have it's getting cheaper and cheaper. However, it does have the disadvantage that the human genome is very large, and therefore it can be quite expensive, especially if you want to look at somatic mutations, because with somatic mutations, you need a certain coverage, so at least 100x coverage to detect those. Therefore, it might just be quite expensive. This is why a lot of people go to whole exome sequencing, because there you focus on the most common regions, let's just say, or the regions which are the ones which encode the proteins and therefore are most likely relevant for the disease. However, whole genome sequencing, again, is getting more and more important due to the non-coding regions. Yeah.
So thanks, Larissa. And with your really great answer, I would like to conclude the Q&A. So now, if you can just jump to the next slide. I would like just, as I said before, to introduce you to our novel Falcon 2 platform. So we have offers when using this kind of platform. And as I said before, especially for RNA sequencing and whole genome sequencing, you send us your samples today, you would have the data back in just 10 working days, which is really fast. So this service is actually available in our Cambridge Sequencing Center, which is equipped with our latest, as I said before, high throughput NovaSeq 6000 sequencers. And can you just go to the, yeah, with this, I would just to thank you all for attending the webinar today. Should you have any further questions or would like to sequence with us, just don't hesitate to contact us through our social networks or through our own website. And just as a reminder, next month we'll have a seminar from Riot, who will talk about DNA sequencing and recent advances. So please do not hesitate to register if you're interested. And with this, I would like to thank Larissa for her brilliant talk, and I would like to thank you all for attending today's webinar. Have a really nice day.